So we have genes, we have about 20,000 genes, which may be 3 or 4 or 5% of our DNA. But the rest is also increasingly viewed as important in regulating those genes and determining how we behave or how a cell will behave. In many respects, when we think about genomes, we think about the whole instruction manual for an individual or an organism. The UK is a great place to do genomics research for a variety of reasons. We have a fantastic history in genomics research. We're just a few miles away from where Watson and Crick discovered the structure of DNA. We're based at the Sanger Institute, which was the single largest contributor to the Human Genome Project, declared as the most wondrous map ever produced. And actually, we should also not lose sight of the fact that the world's dominant sequencing technology was also created by academics just up the road at Cambridge. It's really about having all the pieces of the value chain in one place, and we have that here in a way that only perhaps California and Massachusetts can replicate. If I use one example from academic research that's transitioned into a young company, I would choose the Deciphering Developmental Disorders program. Once that program reached a certain maturity, we spun it out into a company, also based in this centre, to deliver the products and services that stem from early stage research.
So Congenica are primarily focusing on rare hereditary disorders. Rare diseases are those that affect fewer than 1 in 5,000 individuals. Primarily focusing on single gene disorders such as cystic fibrosis or an inherited cancer risk. So the technology that we provide is a clinical decision support tool called Sapientia, and that essentially enables clinicians and scientists to make a diagnosis in patients with rare disease. The way that works is we receive data that's been generated from a laboratory. The data is processed using a number of algorithms. That then is fed into an intuitive, user-friendly interface, which clinicians and scientists can use to filter all of the variation that's been identified and help pinpoint that needle in a haystack that we're looking for to give that patient their diagnosis. So one of the examples where Sapientia has been used to make this diagnosis was a case, a little girl who was 4 months old. She started experiencing seizures. They didn't know why. She'd gone through a whole battery of tests, invasive tests, lumbar punctures. She'd had an MRI scan, and they noticed quite a severe brain abnormality on this MRI scan. So Congenica was sent data from a sequencing reaction for this little girl and both of her parents, and within 15 minutes we had a diagnosis for her. And although the diagnosis wasn't necessarily good news, what we were able to do is find that it had actually been inherited from both of her parents. So they've got that information they can use in the future, and we can possibly test with the family members. And more importantly, we were able to give the prognosis to the child, and she was moved from sort of active management, trying to cure her, trying to give her all of these drugs and treatments, to a much more comfortable palliative care programme to really make sure that she was happy and comfortable in the time that she was here.
For the first time, we can culture all the bacteria out of the human gut and apply genomic technology with absolute rigour by being able to fully sequence isolated bacteria. Our technology differs from fecal transplantation in that, for all its merits, fecal transplantation is messy, it's uncontrolled, it's not a standardized medicine, and it's being tolerated around the world because it's life-saving in C. difficile. But it would be much better to have a highly defined product. And so we're able to identify the bacteria which are transferred during fecal transplantation, which are the ones relevant to the therapeutic effect. And we can then manufacture them under highly controlled conditions without the risk of infection. The gut bacteria affect every aspect of our health and of disease. So whether or not you're a company that's interested in live bacterial products or not, you've got to be interested in what the gut bacteria are doing, how they affect your drug response, how they affect disease. And our technology is the most powerful for identifying the specific bacteria that do that.
Now, what we do is something called nanopore sequencing. So what we do, as the name nanopore implies, we thread a piece of DNA through a very small hole, a And these nanopores exist in nature. To put it in context for you, a single strand of the iconic double-stranded DNA is about 1/10,000th the size of a human hair. And we stream that through a sub-nanometer hole. And as it passes through, because of the size, shape and charge of the 4 bases that are part of DNA, G, T, A, C, we're able to identify them as they slide through this very small hole by using a very simple electronic device that applies a small voltage and measures current as an output. So it's a direct electronic measurement. What that means is we can go from large, complex, huge double fridge freezer size instrumentation in central infrastructures to very small, portable, handheld, decentralized devices. If we look at mortality in intensive care of hospital-acquired or ventilator-acquired pneumonia, it's between 30 and 70%. And the problem is the pneumonia is rapidly mutating and becoming drug resistant. So you need rapid, real-time analysis of the whole of the pneumonia genome. Having a traditional immunoassay diagnostic will tell you known strains of ammonia, but it would not unearth new, rapidly mutating strains. And that is the difference between life and death. And that is a really nice summary of why, alongside traditional diagnostics, genomics will be critical.